So let's do a quick overview of the human brain model. Starting, of course, with the cerebrum. In the cerebrum, we have four main lobes that we can see from this aspect. We have the frontal lobe, which would be this front region up here, to the central sulcus, the parietal lobe towards the back here, the occipital lobe, and down over here where the fold is, this would be the temporal lobe. Now these four lobes coordinate or go along with the bones of the skull that we had seen in chapter 7, so that helps out in terms of the frontal, parietal, temporal, and occipital lobes. There is another lobe, and that is the insula. And the insula is found within this folded region just above the temporal lobe, so I'm going to switch to a different part of the model. If you take a look here, I'm going to remove this top portion of the cerebrum, and what you see is you see a folded in section here. That's where the insula would be located. And you can actually see in both the top of the model and in the bottom of the model. So when this sits on in place, that's where you would have the insula. So that's where you have your cerebral cortex. The cerebrum, we have two hemispheres, so if we turn this and look at a sagittal section, we can see the medial aspect, which is also part of the cerebral cortex. When we have a sagittal section of the human brain, what gets cut through are the regions that are not two separate hemispheres. So this main region here, this is an area of white. This is the corpus callosum, which connects the right and left halves of the brain. Here we have the brain stem, which is cut through the different regions of the brain stem. We have this thickened region here, which is the medulla oblongata. This rounded region, which is the pons. This area here would be the midbrain. Now passing through the midbrain, we have the cerebral aqueduct. Behind the medulla and midbrain where they meet, just in front of the cerebellum, we would have the fourth ventricle. The other aspect, looking this way, we can see the cerebellum here. The cerebellum in humans has two lobes, but it does require a cut through. And when you look at that, I'm going to try and bring that up close. You can see that there is a distinct pattern to the gray and white matter. The gray matter is on the outside, but it has a very distinct branching pattern that is called the arbor vitae. In terms of functional regions and understanding the functional regions, you would have to locate first the central sulcus, which would be located right here, and use that as a guide to determine the different functional regions. So in the human brain, anterior or towards the front, from that we have the precentral gyrus. That precentral gyrus is our primary motor cortex, and in front of it we would have our premotor area or our premotor cortex. That is going to be responsible for the association and the control of patterned responses. The front region here, this is going to be the prefrontal cortex. The prefrontal cortex is where we would see things that would be along the lines of personality, cognition, intellect, reasoning. Okay, would be housed in the prefrontal cortex. In the occipital lobe over here, at the very point of the occipital lobe, is where we have our primary visual cortex. And the majority of the primary visual cortex actually is going to be coming up here on the median surface through this very distinct sulcus. When you're looking at the median surface as well, you can see this delineation here. This sulcus here, this is the delineation between the parietal lobe and the occipital lobe. Alright, so let's just go through and take a quick look at the preserved sheep brain. One of the first things that you should notice is that the sheep brain is much more linear in organization than the human brain. So we have the cerebrum and the brainstem in pretty much a straight line with the spinal cord. And this has to do with the fact that sheep, of course, walk on four legs with their face forward and then they're straight back. So it's very different than what we see in humans. Humans have basically a 90-degree bend between their brain stem and their cerebrum. Okay. So we'll start off with this. This is an intact sheep brain. We have this large area here, two hemispheres. Those will be the cerebral hemispheres. This outer portion would be the cerebral cortex. You can see that these are hemispheres, and there's actually a space between them. This would be the longitudinal fissure, okay, where we have the two different halves, the two different hemispheres. Looking from a side view, here is the cerebrum. Back over here we have the cerebellum. Okay. The cerebellum on the sheep brain is really one large lobe, as opposed to what we see on the humans, where we have two distinct lobes. Now, turning this to look at the inferior aspect is now facing towards the camera. We see here the olfactory bulb. Right? The olfactory bulb is towards the most anterior part of the cerebrum. Right? Then we have the olfactory tract, which brings in those sensory neurons that are involved in the sense of smell or olfaction. This is the first cranial nerve, so cranial nerve number 1. Here we see this distinct X. This is the optic chiasm. Right? This crossing, what you can imagine is from this your optic nerves would come out on either side and attach to the eyes. These optic nerves, which is the cranial nerve number 2, would then pass in through the thalamus where they would be routed. The other structures that you can see from this aspect very clearly, you have the rounded portion of the pons, the thick region going into the spinal cord, which is the medulla oblongata, right, and then in between we have the midbrain. Also obvious are the mammillary bodies, which would be the portions of the hypothalamus that are able to be seen in this view. Now here we also have another intact sheep brain. The difference is this still has the dura mater, which is the outermost of the meninges, intact. I've cut a little section away so we can flip it back. This is the consistency of like wax paper or saran wrap. You can see that it covers the brain. This would form pouches, separate pouches around each of the cerebral hemispheres. If we take a look at the cerebellum, we would see the cerebellum is also surrounded with dura mater. Once again, this is keeping the cerebral spinal fluid in, circulating around the brain. If we look at the inferior aspect, there are areas where you do not have dura mater. One of the very clear areas is where we have the olfactory bulb. In terms of the sense of smell, the olfactory bulb needs to not be blocked by the dura mater so that actual olfactory filaments can pass down into the nasal passageway. Now this next item, this is a sheep brain that has been cut in a sagittal section. The sagittal section allows us to see the medial surface of the cerebral cortex. Right? So the cerebrum, the cortex actually folds in. The area that has been cut through, that is the corpus callosum. The corpus callosum is an area of white, which is connecting the left and right halves of the brain, of the cerebrum specifically. There is a space in here. I'm inserting the probe into it and up. That would be one of the ventricles, which in a living brain would be full of cerebrospinal fluid. Cut through here as well, you can now see this very clear round area would be the cut through of the optic chiasm. If we turn it on its side, you can see where the optic nerve would be. That's a very important landmark. You would also see cut through, this is the cerebellum. It has this kind of tree-like pattern called the arbor vitae or arbor vitae. This is a pattern of white, where you can see the difference between the white matter and the gray matter. We also have the pons, the medulla, spinal cord. These are all cut through. There are two hemispheres in the cerebrum, but the brain stem is singular, so it has to be cut through when we're looking at this sagittal section. So those are the important structures that you may see on the sheep brain dissection.